This is a very dear to our hearts workshop that we were wanting to do for a long time. We were waiting to have enough information to fill some of the gaps that the Tara Microbiome expedition wanted to fill, and to have a clear overview to what was achieved on the expedition and what the challenges are for today. So I just want to welcome you all to the Universidad de Concepción headquarters here in Santiago. It's a real pleasure to have all of you here, and I will invite Alejandro to come with me and say hi to you all as we are co-coordinators of this event and the CEODOS program.
I have 30 minutes of no.
He will fill in the 25 minutes remaining.
Yes. No, first, I don't know why I am here. I am grandfather from Saturday, so the first girl in my family. This is very important for everybody.
You can show the picture, yeah.
Loudly?
He will show the picture.
I will show the picture. We are so, how to say, proud. No, but thanks everybody for being here. We have been discussing for many years to be together. We created this consortium CEODOS X years ago. We were 8 centers, at least we are 4 here.
Half of us.
So the first 4 in the list here, CMM, CGR, COPAS, and INRIA. And the idea was to create kind of multidisciplinary teams to put together to analyze data from the Tara expedition in Chile. Together with us, we discussed a lot with Daniele and people from AtlantEco, and maybe this is the first time we can be the host here. Daniele was the host, at least for me, in Portugal. It was a very, very important, interesting discussion. I learned a lot, and I think that this workshop was created to learn a lot again. All of us, your experience, our experience. So thank you very much for being here. 15 people crossed the, how we say, the charco. Some just crossed the Andes, the other crossed the oceans and the complete continent. So thank you very much. We know this is a big concern today. We discussed with Olivier at some point about and yesterday in the mountains about the CO2, yes, what to do to compensate, yes.
Yeah, so please, when you return home, use the bike as much as possible to compensate for this trip.
Without engine, yes?
Yeah.
A good one. So thank you very much. We hope today will be a nice day. Today is more Science to Policy Day. For the moment, we are only the science. The Science to Policy will start later. There will be 2 talks from people in France, from OFB and FFM. André will tell us. André he has a seat in the United Nations for the discussion about the ocean. So André will tell us what are the feelings in those forums about what science can be provided to go ahead with laws of the sea, etc. And this afternoon we'll have some authorities in a round table discussing what is their view about the science we can provide. So from 3 ministers in Chile, Minister of Environment, Science, that now they have someone that is in charge of foreign affairs, in some sense, and also from the group in the Minister of Foreign Affairs that is discussing in those forums about the ocean. Yeah, so this is today, and after, to those that will go to Valparaiso in the vans, just follow me. We will try to see where the vans are parked.
Usually at the front.
Yes.
Don't worry.
I don't know if I was afraid about the traffic in Providence. I am always afraid anyway. Okay, so thank you for being here, and I hope you have a very nice and productive week, and overall to create a lot of links and collaborations for the future. Yes?
Yeah, I mean, I want to remember and remind you all that not everybody here was directly involved in the actual expedition. Some of you are touching the results, from on the side. Some of you are interested in the implications of what we achieved in Terra microbiomes. And this is a huge undertaking. It's a huge expedition, a lot of collaboration that need to be continued over time and fed by the scientific results and interaction that we can create in workshops such as this one. So it's very important for us to have your feedbacks, your questions, and actually to talk about what the possibilities are. For the afternoon, as Alejandro said, there will be the science to policy part of the workshop, which is also very important because the implications of what Tara Microbiomes wanted to achieve have global objectives and touches directly what the IPCC and the ONU want to the message that they want to pass to society for 2030 and beyond. So we have kind of a responsibility here that we are happy to take over, and to fulfill. So again, welcome on behalf of us all, and let's kick off this week of Tare Microbiome and beyond. Well, you have the program, so what comes
Daniele, I
next?
think you start. Yes. So the first talk is from Daniele Giudicone. Daniele is a world leader in.
Do you know this?
I have to say that. No, anyway, Daniele is the coordinator of AtlantEco expedition, the second part of Mission Microbiome, and he really has been working a lot for the last year. He's one of the inspirators also of the expedition in Chile, yes, in CEODOS, together with Damian, Colomban, and André. And so we are really happy you're here. You could do the travel, and his talk is about the challenge and the expedition in the Atlantic. We will continue after with the Pacific side.
So thank you for the invitation, and it's always a pleasure to be here. And my talk today will be basically introducing you to AtlantEco, which is actually a European project. So, and then I will move to discuss what we did for the expedition specifically, which is one of the expeditions we have been doing in the context of this European project. So, a European project, as you probably know, so in Europe we have this system of funding across different countries on specific issues, and in this case it was the establishing the health status of the Atlantic. And what is specific of this call, which is a little bit of a geopolitical call, is that Brazilians and South Africans colleagues were eligible for funding. So what is really nice on this occasion that we can really cooperate and collaborate and work together. And I'm really happy that now we have also Chile, so we can extend this collaboration, and also Argentina, which are in it. So one of the reasons I'm really happy to be here is that to try to help to create this network in South America, which is one of the aims of this project. So very quickly, the oceans, you know, it's our incredible place. I'm a physical oceanographer, just to clarify. I'm not a biologist, not an ecologist. I'm a physicist. And so, but also we stress the oceans a lot. Sorry, let me see. As you know, because of different stressors, it regulates the climate. So it participates in the regulation of climates, as you know, regulating the level of CO2 in the atmosphere, but also redistributing the heat. So for instance, the ocean has taken 90% of the heat, the excess heat due to climate change. It provides proteins to humans, and that sense is still very important, especially regionally. And we love it, but we stress it. So there are many issues, from acidifications, overfishing, microplastics, and so on and so forth. So in this context, it's very important that we set up this sort of virtual loop where we inform policy, and at the same time we activate a sort of loop where governing and acting is also allow us to improve our observations and mapping, which then, using science, is getting a better understanding, which in terms allow us to predict the response to stressors, and with this prediction we can inform the policymakers. So this is, we are supposed to contribute to this virtual loop. Is not easy because we still don't know many processes acting in the oceans, as I will argue later on. But at least we are trying. And I'm really happy that today we have almost all the components of this loop in this room. So the project focuses, so the general question is what is the health status of the Atlantic Ocean, and how can we predict the response to climate change for the future. We decided to focus on the ocean microbiome, which is basically the soup of invisible organisms that lives in the oceans. And the reason is that they form more than 80% of the biomass of the water column. They provide a lot of services to society because they contribute to carbon sequestration, for instance. They are the base of the trophic web in the ocean, so supports also the production of proteins. They also channel pollutants, and so there are many good reasons for society to have more information of how the ocean microbiome works. What is the ocean microbiome? A little bit more in detail. So it's a combination of viruses, bacteria, unicellular organisms, and depending on the definition, we include also small animals. What is amazing is that we cover a huge range of sizes and for these organisms. And so the main point, at least for me, is this. So the point is that we want to predict the response of a system, but if you look more carefully to the system is made of 3 complex systems which are coupled together. So the ocean as a fluid is already a complex system. Okay, chaos has been found analyzing the coupling between atmosphere and ocean. The organism itself themselves are complex organisms because of complexity of the biology within the organism. And then we have the complexity of the web of interactions, which are not necessarily prey-predators. There can be positive interactions. But anyway, we have multiple thousands of actors which interact in the ocean. Each one has a complex biology, and they all live in a complex fluid. Okay, so this is the challenge, the conceptual challenge I believe we have. And in fact, if you look at the details, the ocean physics alone is really already very complex, which means it's very difficult to produce predictions because we do not know yet exactly how gas exchange, for instance, between the atmosphere and the ocean is occurring, depending on the wind side, etc., etc. So the point is that instead of simplifying the system, as we have done for many decades, we have to phase the complexity, okay? Try to extract information and knowledge out of heterogeneous data and trying to reduce the complexity to get assessment, understand, and prediction. One way to see this also is this, as we did in Tara Oceans, is to see the different level of complexity in terms of spatial scales and nesting level of complexity, where we go from the biology of the organism up to the web, up to the biogeochemical cycles at the global level. One step change in this way to understand the processes has been recently the introduction of genomics as a tool to assess and understand. Okay, this is mostly coming from human health research, and basically using genomics, so sequencing DNA or RNA of the different organisms in the environment allow us to do taxonomy, so understanding who is there, even if it's too small to be seen with a microscope, allow us to understand what is the potential functional response of the organism through the analysis of DNA, and then tell us what was the metabolic state of the organism in the environment that day we sampled using analyzing the gene that were actually active in that place that day. So Tara Oceans, which was an expedition we had done more than 10 years ago, paved the way to introduce systematically this approach into ecology, marine ecology, because we had this expedition all around the world where we sequenced the RNA and DNA for all these series of organisms, and this allowed us to do many first studies, and for me the important point today here is that this allowed us to learn how to work with this data. These are very complicated data because a gigabyte of information for each station, which is completely different than before, where we use temperature, which is one number, salinity, one number, nutrients levels, maybe chlorophyll, maybe photosynthetic responses, these kind of things. This was marine ecology before. Now, for each station, we have a huge amount of information, which is great, but at the same time, it poses several challenges, and which is one of the main good reasons why we have to work together, okay? Because we don't know yet how to work with this data completely. The other point, I'm a physical oceanographer, this is I care particular to this point, is that let's not forget we are in a fluid, which means that everything is on the move, and the impact of the fluid goes from the large-scale gradient, okay, with the currents, to the mesoscale. We call this mesoscale, so kilometer scale phenomena. So basically the microbiome is actually distributed along patches of this size if we zoom in here. But then if you go at the level of the organism sites, we see the interactions between the different organisms are mediated by a fluid again. So we have to keep this in mind, which means that many theoretical conceptual advances we get in ecology, since they are coming from terrestrial ecology, maybe we have to throw them in the trash and think to new ones, okay? But anyway, this is really important. And in practice means that so this is a sketch of a community. So a community travels all the time, right? And then experiences different processes. And the main point is that you cannot consume time without consuming space, okay? So if you want to look at the dynamics, which means you want to look at the system in times, how it responds to perturbations, actually you are also consuming space, you're moving. So you cannot decouple space and time, and the way you have to do it is it's still unclear. It is a factor reason, as I will argue later on, that the need of also process studies dedicated to understand this kind of coupling. I give you an example of why this is really important now, so to understand the seascape. Okay, this is a paper a few days ago. So basically, as you know, now there are billions of dollars to try to catch the CO2 from the atmosphere and to put that somewhere to reduce the amount of CO2 in the atmosphere. So there are a lot of people now is thinking about this carbon dioxide removal processes. So it could be industrial, could be other approach. And one approach many are proposing is to rise the alkalinity of the oceans or to fertilize the ocean in specific places, trying to create an imbalance of CO2 between the atmosphere and the ocean so that there is a sequestration, okay? It's an artificial form. There are billions of dollars of investment in this direction, okay? But the point is, this is an example of a nice paper. So they try to reduce it, to create an imbalance. So they reduce the CO2 in the ocean in the first level, in the first meters, in 3 places in the ocean. So this is Iceland, this is Brazil, this is Fortaleza, and this is in the Pacific. What is interesting to see that after 1 year, the imbalance between the atmosphere and the ocean, so the sequestration, the active sequestration, is completely different depending where you did it, okay? And this goes on for 5 years. But most importantly, depending on the place where you do it, you can have that either it's a big success, so you actually have 100% efficiency in the sequestration. But if you put this in Iceland, your sequestration efficiency is only 50%, which means that the way the fluid redistributes the perturbation, the artificial perturbation done to create this sequestration, is crucial to then have an efficiency. And consider that the idea of this paper is that we can try to map the seascape and associate an amount of dollars that you can compensate with for carbon utilization. So another challenge we have is that we have to connect our understanding of the biology of the organism with the way we predict the response of the biochemistry of the oceans to a climate change, okay? So basically, when we do this, the IPCC prediction or these kind of things that we use for then regulate the society in terms of emission, basically use very simplified numerical models of the biochemistry of the ocean, but actually these are few state variables, while there is a huge complexity in terms of biology, and we still don't know how to to fill the steps here. Okay, so this is another good reason why we have to understand more biology, understand more how then all this is translating into rates of transfer of matter and energy, and these are all open challenges. Another comment I want to make, still at the beginning, is that after the COVID, now there is a clear idea that we have to protect the health of humans, we have to care about the health of the entire environment in which we live. Okay, so COVID was coming from an animal, we don't know yet where, and now so the World Health Organization pushes for a One Health view, which means to consider at the same time the human health, the health of the animals we eat, and in general the environment, and most importantly to use the same approaches. So you introduce in genomics, proteomics, metabolomics, in all the three fields, in a way that we can then have information for predicting, for instance, pandemics, new pandemics. So in this context, we had the occasion to have funding from the European Union. It's to have this project. So AtlantEco is collecting, basically started as a small group of people that did Tara Oceans before. So we're thinking about how we can try to work together and introduce the concepts on omics, using omics into oceanography. And so we were lucky, so we formed a large team, 36 partners, so many Europeans, 3 partners from South Africa and 4 from Brazil. And we profited of this occasion, and we were able to get the funding for 5 years. It's a big project, so 36 partners, it's more than 100 people working on that. It's 11 million, the funding, million euros. The project is around, as I will tell you more in details, is basically around 3 pillars. So one is focusing on the microbiome, introducing systematically in oceanography the approach we had in Tara Ocean. There is an aspect related to trying to understand not the plastics in the ocean itself, the distribution, but trying to understand who is living on these plastics, because it's a new substrate we are introducing into the oceans, right? So basically species that were not able to live without a substrate, now they can live in the middle of the ocean, while before they can only live on the rocks on the shore. Okay, so we are creating a new substrate, and so it's very useful to use the approach, the genomic approach that we use for the microbiome, to try to understand what is living in the plastisphere, which is let's say the ecology happening around pieces of plastics in the ocean. All these in the framework of the seascape. So keeping in mind the seascape view is important, and the connectivity, the fact we are in a fluid, is crucial to move forward. So the idea is to try to develop a new holistic framework that will be useful for managing health and services, and provide knowledge-based resources, engaging also in industry, policy, and citizens, which is not obvious to do, and also to exchange capacity across the different countries, okay? And the idea is not really, we want to cooperate. So the idea is that we want to work together. So not helicopter science as we do. We go in a place, we take the data, and we do the science in Europe. No, the idea to work together. And as I will argue later on, let's empower also the countries that in the future they can be totally independent, okay? Because there is a problem here, clearly, with this approach. These approaches are very costly, really costly, and they need a lot of infrastructures from sequencing centers, bioinformatics team, etc. So it's not really easy to do this. And also, as I will argue later on, we actually have probably, because of this project, to rethink the way we do this approach, because sometimes it's too complex or too costly. So it's very nice to have a very large team. We work together, we try to cooperate. I'm not the boss, I'm just the coordinator, okay? I just have to have the thing working and move forward. And there are people from many, many countries working on this. So the first pillar is to focus on the microbiome, this invisible majority in the ocean, and where we basically have improved the Tara Oceans protocols, trying to have the same approach and to have this approach systematically used in many oceanographic cruises, as many as possible, and with the idea to and this is very important for the project, to combine together also with new models, new numerical models of the oceans, new conceptual views from bioinformatics, trying to build pieces of what could be a future approach, which is fully multidisciplinary, where this dialogue is basically permanent. So basically, you decide where to go for the next expedition because we had some insights from the previous data, and so on and so forth. The second point, of course, I will not spend more time here on that. It's the seascape. So we have groups working in the project looking at the Lagrangian, so-called Lagrangian dispersion in the ocean, and we try to connect this dispersion with the processes acting on the ecosystem. I just want to show you here quickly that if you look at the stressors for the South African region or the ocean, the stressor for the end of the decade, the change in circulation, in the ocean circulation, okay, it's the highest stressor which is predicted from the IPCC. Okay, just to give you an idea how changing currents is also a stressor because you change the pattern of the species. I don't have time to. So microplastics, what we are trying to do is basically, as I told you, that the cycling of microplastics is complex, but our interest is basically to see the coupling with marine life, but above all, trying to use genomics to understand who is living on the plastic. So bacteria, could be diatoms, could be protists, and other kind of things. And to use this information to better understand if there are toxic species, for instance, living on the plastics, how this plastic, if it is eaten by an organism, what is the impact not only of the plastics, but also the organisms living on the plastics on this thing, how the weathering is impacted, and so on and so forth. We have, I'm not going to the details, but the project is very complex. So we have 10 work packages. We have some also capacity under outreach. And we have also, in addition to what I described until now, people working trying to understand the drivers of this ecosystem. So which means to try to understand in the last decades, for instance, which were the processes that created changes in the ecosystem. Could be atmospheric processes, could be direct human intervention, and so on and so forth. We have also, we are going to try to push also the methods and using new approaches like let's say improved approaches like network approaches. We are working also to augment our capacity to observe, so we have some technological development on sensors and so on and so forth. So it's a big project. So one of the axes is improving ecosystem description, the ecosystem function, which basically at the moment is mostly collecting all the existing data at the global level. This thanks to Ugo here and others, and we are producing probably what is the first global database of the genomics of the microbiome of the oceans. Okay, it has been released, the first version, a few months ago, Ugo, no? And it's the first time there is basically all the genomic data available have been redigested with a single unique pipeline, and these are now available, and everyone can then do the studies they want, for instance, on the biogeography of the ocean, and this is available. We want to, sorry for the slide, so again, enhance and innovate, so we are trying to use a different approach. One approach is introducing artificial intelligence. We really didn't start yet, actually, but is also to trying to improve the biogeochemical models of the ocean that are used by IPCC, using the gene expression in different regions as it is connected to the biogeochemical status of the ocean. And then the analysis of the drivers I was mentioning before, which drivers is not only the drivers that change the ocean because of like atmospheric changes, like North Atlantic oscillations, but also trying to understand the human drivers, and then to try to develop, working with the two economists. We have two groups working from economics that they are actually trying to use our information to improve the predictions of the response of the ocean to changes in the societies all around the basin. It's a very big challenge. It's not obvious. And then we have this effort in trying to have sharing capacity, etc., etc. The main objective of the project would be to basically to end up the project with a roadmap for the future, okay? So having tested different approaches, having worked together with putting together different communities, how do we then move forward in the future into integrate this integrated these approaches. I just want to mention one. I mean, it's a long list. I don't want to detail this, but one challenge we have, at least in Europe now, is the building the so-called digital twins of the system. So basically, there is this idea that we can produce a copy of our real natural system in a computer, okay, and then use it to try to understand how it would respond, for instance, to climate change, this kind of thing. It's much beyond simple modeling, okay? What is fascinating here is that this coming from physics and engineering, okay, twin model of an airplane, for instance, but for biology, no one knows how to do a digital twin of the ocean, okay? No idea. The complexity is too high. You have millions of state variables, okay? And I think conceptually, in these days, this is one of the most interesting challenges we have to contribute to. Then we work on a series of ecosystem services that are provided by the microbiomes. And we have also some case studies working in the private companies, or for instance, we are now working in South Africa with the company that mine diamonds along the coast, trying to reduce the impact of this mining of diamonds. So let me get to the part on the expedition. So one important part of the project is try to introduce regularly these approaches into oceanographic cruises, but also into monitoring, coastal monitoring observing systems. For this aim, we empowered, first of all, we did a series of, and we are doing a series of flagship cruises of the project with different systems, one with Tara, because I will detail in a minute, but we also other sailing boats. Now we are mostly working with the Modelamiento in Brazil, and Paula is here, maybe you can tell something later on that. And we have many other cruises which are third-party cruises that have been going all around the Atlantic in these 2 years, and we empower them with our protocols. We are empowering the mining stations all around the basin, trying to pass these protocols for monitoring, and also working with people at Roscoff, with Colomban de Vargas, to try to have citizens using these protocols to map the status of the microbiome. And then we have a training and literacy program we are developing with UNESCO. And one of the most important points is that we want, as I was saying at the beginning, we want to have a permanent change in the way we cooperate. Okay, so that's why one aim of the project, which is not easy at all to reach, but we are trying to empower the sequencing centers in South Africa and Brazil to pass the protocols with respect to the Genoscope, which is the reference institute for. And the idea is really to let them to be totally independent from Europe in the future for these kind of exercises. And my hope is that here today, in these days, having people from Argentina, of course from Chile, that this story we can develop further this idea. Now let me go to finally. Sorry for that, but I wanted to give you an idea of the context. Okay, so our expedition, the expedition we're going to talk today, is happening in this wider context, which is really important. Okay, so Mission Microbiomes is this expedition we have been organizing together with you in Chile. I will present the Atlantic part quickly. So the expedition was this one. So we started from Lorient, then we went down to Chile. Then there was a Chilean part at the beginning, and Camila will talk about it later on. And then we did the Atlantic part of it. Okay, where it was a tough experience because of COVID, because an expedition is always complicated. We had to deal with things like permits for sampling, and thanks to people like Federico, we were able to overcome Hugo, to overcome this huge problem. And but overall, even if it was very complicated, it was a great success. So we have more than 150 stations, full stations, with advanced protocols, standardized protocols in regions, in coastal regions, which have never been sampled before, and which as ecosystem services providers are much more important actually than the pelagic regions, right? Because it's where the people live. So first of all, we produce an handbook with standards and best practices. Stéphane Peson was able to synthesize and build upon the experience we had in Terra Oceans, introducing further elements on it. And specifically, we added to this an eDNA approach, which means to sample metabarcoding, where basically you try to look for traces of DNA or whatever is in the water. It can be whales, can be fishes, okay? So you are going beyond the microbiome. So basically you sequence what the pieces of the organism, and so it's a way that a lot of people now is using to trace and characterize the trophic web. We introduced more than before single-cell genomics. We sampled turbulence and sophisticated physics for the sailing boat. Trace elements, isotopes for nitrate and nitrification, long reads, sequencing, nanopore PacBio, microplastics I mentioned, and then we introduce also regularly metaproteomics and metabolomics as tools, because the idea is that we are to go beyond the gene expression as a way to characterize the metabolic state, because gene expression is only the first step, right? What is important are the metabolites, are the proteins that are produced by the organ. So now we are able, thanks to new protocols, to get directly to this information, and it's interesting to have this together with standard genomics in the same place for hundreds of stations. This will be very valuable to compare the different approach. So this was the expedition going across very different biological regions. So in green, this is the level of chlorophyll in the water, so basically biomass in a way. And the expedition was, as I will mention in a second, was divided into process studies, okay? This because again, the idea is we want to understand the seascape, so instead of doing a regular survey, we say that we organize working groups together. We had workshops before to try. Sorry, problem? Okay. Now, so we had a lot of discussion. So we split the expedition into process studies on the different regions. So first we formed a group, then some groups, then we discussed the main questions and the state of the art of the understanding and knowledge, and then we designed sampling plans which were addressed to improve our understanding of the processes occurring there. Okay, could be the plume, how the plume of the Amazon River is dispersed in the ocean here. So it's quite amazing that your river coming here, that the impact of the river is over thousands of kilometers in the ocean. So we're trying to understand why, how, the bloom dynamics with Federico and others in this very dynamical and very rich region in Patagonia, and so on and so forth. I will tell you more in a minute. So process studies means also that we added, depending on the cases, added things like using floats, using SAR or other kind of satellite real-time following of the system, so that the sampling was really, really dynamic, okay? No transects, basically. So I focused on oceanographic planning. Another point is that we formed this working group, and the working groups were chaired by younger people. We try to get the old people away from directing and managing. So there was an excellent work done, for instance, by Federico, by Paula in Brazil, etc., which was really rewarding. We dedicated each process studies to a female scientist from the country, neighboring country, okay? Which was something we decided from the beginning. It was really, really nice to see how this developed and how was the reaction from the different groups. I'm sorry that in Chile I didn't succeed in convincing you to do the same. I don't know why, but anyway, I can tell you that in most of the other legs, everyone has a name of a person, is always dedicated to someone. And now in the future publication, the experiments will have the name of the female scientists. Okay, so it will be the experiments, Shackleton, for instance, for South Africa, and so on and so forth. So standard baseline. So when possible, we did synergies with other cruises. So we tried to contact oceanographers that were in the region. In the same time, try a coordination, like as we did in the Weddell Sea, or as thanks to Federico, we even had boats at the same time with the same protocols, going around in the same region, trying to increase the capacity to cover the problem. So very quickly. So the question we address is the impact of the Amazon River in the trying to have an understanding of why the plume is so wide and what are the biochemical and biological responses to the strong injection of nutrients and plastics in the ocean. We did this experiment with our Argentinian colleagues and others from Weizmann on this region of Patagonia. So we are this is the Malvinas front, which is very active, and where we try to see the impact of viruses on coccolithophores bloom. We did an experiment, a process studies trying to address a mesoscale feature while crossing the Atlantic. In the Weddell Sea, we focused on basically what is the biological pump over there, because as you probably know, all around Antarctica we have a huge push. It's a sort of huge fountain, Antarctica, right? Deep waters are pushed up by the winds. They are very rich in nutrients except iron. And so the biological pump there of CO2 and drawdown of CO2 is very important, but we don't yet the biological components of that. More on that, we had the pleasure, this was really a pleasure for me because it was very challenging, but also we asked the question, is an iceberg, given that you have a lot of nutrients except iron, is an iceberg coming from land, so with some land attached to it, rich in iron, is these fissures releasing iron in the ocean, so promoting a response by the ocean, fertilizing the ocean. So we chased for 2 weeks an iceberg from satellite, and with the boat was sampling all around. So these are more on the Argentinian experience. So we had this Hussein sailing boat, which was organized by Federico and the others, which was sampling about at the same time of Tara, so they were nicely complementing. And what is really moving for me that this is actually the same boat as it's the Atlantis from Woodsole, okay? So that is most of the biological and physical oceanography we know was actually discovered on this boat in the 30s, in the 20s and the 30s in Woodsole. So the mesoscale was discovered doing transect in front of Woodsole with this boat. The trophic web, many elements of the trophic web, the fishery dynamics were discovered with this boat. So it's really nice that almost 8 years after, two very important boats for the history of science were doing something together. It's really something moving for me. Anyway, so the greater upwellings is another question. So we targeted, thanks to our South Africans, colleagues that were able to involve Namibians, people from colleagues from Angola, from Congo. We were able to map one of the most important nutrient-rich upwelling, which is the Benguela upwelling, and then also at the end of the expedition we targeted the Senegal upwelling, even if it was not the upwelling season. So this is a case of synergy. So then we enabled a French oceanographic cruise that were going in the same region 6 months after, so during the upwelling region, with the same protocol, so now we can compare the systems. To finish, we also targeted the rivers. I told you about the Amazon River, but also we did some transect in the Congo River, which is the main source of iron for the entire South Atlantic. I'm concluding with 2 comments here, practical comments. So I think now we have to work together with this data. They are fantastic data, but still we have a lot of challenges now in terms of coordinating on, if we want, to things like, for instance, something I think we have to discuss in these days, that what are the bioinformatic pipelines you want to use, because if you use the same, it's better, so we can merge the data and possibly merge the data with the Tara Oceans data so that we have a huge new data set that we can explore. And also if you want to contribute to the intercalibration exercise that you and others are organizing to try to let's check if the different pipelines in different sequencing centers are well organized. In terms of data analysis, we have the physical data analysis ongoing. I have a postdoc on that. The chemistry, still we have to organize it, so nutrients, HPLC, and these kind of things. For the sequencing, we are already sequencing some of the station. About 1/3 of the station will be sequenced in the next weeks or months, and where we focus on 3 process studies. then the sequencing it will be going soon, I hope, in Brazil and South Africa too. So we are quite hopeful that we soon have data. And so what we are going to do in the next months, given the data are starting to come, is to form working groups on the different process studies to start working really together to understand the processes, but also it will be interesting to discuss if we want to have a joint group on general questions where we put all the data together, we try to address a question which is independent of the specific local place where we sample the data. The other challenge is that I'm really fond of is trying to have an impact on the policy. To me, what was amazing of directing this experience was also the human side. We learned a lot, okay? Confronting with our Brazilian colleagues, with people from Angola, from Suriname, from Argentina, from Chile. Many countries, and each time was it's a lot of time of discussions and confronting the different systems, etc., etc. So I think we learn a lot in terms of how we do cooperation. And I invite all of you to contribute to do together a policy brief for the EU to try to give a feedback on them, okay? And so they are the funding members, but you know, I realize, for instance, that you have to talk with the people you want to cooperate before writing the proposal, right? Not after. Because then you have a better proposal. You have to design a better plan, okay? Which is really going to help for the cooperation. And then there is an important point here, is that, so this was based, this geopolitical call was based on what's called the Belen Declaration, which states that Europe, Brazil, and South Africa wants to work together on the Atlantic Ocean. Since then, there was a new declaration where the Galway group, so the EU, Canada, and US, which already cooperated, joined this other southern group, and then Argentina was added, Morocco, Cabo Verde, I don't know about Chile, and so now they have a new context, and it will be important, I think, to lobby together to have new fundings to have a wider group for the future. Thank you very much. Sorry if I was too long.
We will have time for questions during the week, but I think now we can open quite many questions or comments from Daniele if you want. Was so clear. Now, do you have a time frame, Daniele, on some of the things that you...
For the analysis,
yes, any kind of analysis?
No, as I was mentioning to you, the state of the art is that the physical data, the analysis are ongoing, so we should have soon, validation, calibration, etc. of CTDs, turbulence profiling, the online system, and so on and so forth. We have transferred the data for the chemistry. Now we have found also the money, maybe, and so I hope in a few months we'll have the chemical data. For the genomics, we get some funding, so we had the discussion. We have some priority stations will be sequenced. I don't know actually the status in Brazil at the moment.
We are working on the comparison of the sequencing servers before we sequence, but certainly during this year it's going to be finished. The project goes until 2025, so.
Yeah. And you're planning to go from prokaryotes or viruses to eukaryotes?
Yeah, we're going to do metagenomics.
So all what was mentioned here is in the pool?
Yes. Well, the part of the Brazilian will be sequenced in Brazil, and the part of the African coast will be sequenced in South Africa. That's why we need this inter-comparison of sequences.
This decision was taken during the project, during the expedition. We realized that it was better, right? So it was nothing.
We did also an important step, a difficult step, which was shipping the samples from Brazil to Europe for the chemical analysis. So it takes a little bit of time and bureaucracy as well.
Yes, Camila.
Are any of the programs, the local programs that you worked on, going to be maintained over time, like time series or?
It's a very good question, actually. Yes, at least for Benguela, yes. So because in the meantime, I didn't have time to give you the details, but in South Africa, we empowered also the local ships, the local vessels. So now they have the filtering system, and so they are going to have this regular transect for monitoring the Benguela quelling using this approach. This at least. I don't know about Brazil. We have to discuss yet, but that's yeah.
Okay, so thank you, Daniele.
Thank you.
I'll talk tomorrow, so we don't want to be. No, not tomorrow. So it's Thursday. Next talk is by Camila Fernandez. She's a researcher, director at one of the centers in CEODOS, Copas Coastal at the University of Concepcion. She's also researchers at CNRS at Toulouse in France. So she's one of the coordinators of the CEODOS expedition, and the idea today is to give some ideas of the expedition, but also some results, I think, yes, preliminary results that start to be there.
Some raw data. So it will be also interesting to see what comes next. So thank you again, Daniele. Thank you so much for your talk, because you spared me a lot of time explaining the context and why it's important to focus on the microbiome and the biological pump. So, okay. So once upon a time, there was this expedition centered along the marine microbiome. It was a joint effort between many institutions, but mostly pushed by the Tara Foundation and the AtlantEco program. So Alejandro was very right to say that Daniele and the team in Tara were pushing it for this expedition in a very particular time in our history. So the first leg of the expedition was the Chilean part. They sailed against all odds in 2020, December 2020, from Lorient, in a time where the whole world was in quarantine. We were in lockdown, complete in Chile. Almost anybody couldn't come in the country or go out of the country, not to say ships and scientific materials for sampling microbes, and try to explain that to the authorities. We want to sample microbes in a time of COVID. So that was a challenge, but the idea was to test a little bit all the protocols and the focus of the expedition during this first leg. The challenges were enormous because, as I said, we were in quarantine, so it was virtually impossible to even conceive that the ship Tara was actually going to be able to go down, and against all odds, they did. We received the ship in Punta Arenas in February 2021, and then sailed north with it for 2 months and a half. The whole thing lasted for 4 months in our shores, and the working group that was created especially for this first leg of the expedition was called CEODOS Chile. And so my role today is to describe to you the objectives of this consortium, a little bit of history about why we wanted to do this expedition, what were our scientific questions and objectives, and hopefully present a little bit of results and data that came out of it, along with the perspective and the work that we need to do yet. So yeah. So this is an image of Tara in Patagonia. This was one of the first stations they did. The idea of the whole thing was to test the hypothesis that we can actually map the biological pump along the Chilean coast. And why along the Chilean coast? We're going to see in a few seconds. But this is for presenting the actual track. As Daniele said, they sailed down, and the expedition started here with 4 stops along the way, along the Chilean coast. As you may or may not know, Chile is more than 5,000 kilometers long and is a very thin country, so it's basically water. And there's a lot of features that characterize this coast that we wanted to study in detail with a very limited amount of resources, time, and people. Basically, Tara is a 36-meter long ship. It was the only one working in our waters during this time. There was nobody else in the water. We will have no possibility of having a second or third ship working with us. And the whole crew was 14 people, which meant that in each of these legs, we could embark only 2 persons from our team. So together with the European scientists, AtlantEco scientists, and CEODOS scientists, we managed to create a scientific program for specific conditions on the Chilean coast that was designed to map as much as possible the genetic diversity and the biogeochemical dynamics of the biological pump along Chile. Of course, this was an administrative challenge, and we had a lot of support from the Ministry of Science, Marine Environment, Foreign Affairs, Defense. There was a huge undertaking from the communicational point of view because nobody could go in the ship or out. And for Tara expeditions, outreach, education, and the communication of the way we do science for exploring and understanding is very, very important. So for us, and yeah, hi, Andres. You're in the picture here. So thank you so much for coming. The former Minister of Science is here, and he visited the ship right before the end of the Chilean leg. So a lot of support and believing in this project came from the Ministry of Science, and we are forever grateful for this because if not, the expedition probably couldn't go forth. So a lot of logistics and learning how to do this kind of expedition was required from the science team and from the administrative team as well. We made a lot of outreach efforts, and we touched almost 2,000 students along the way in 4 months. And we sampled some very specific features as well. So the whole thing is based on microbes and microbiomes and the role they have in the biogeochemical cycles in the ocean and the coupling with the atmosphere. So for us, the main question was, how is the biological pump behaving along the coast of Chile? We know that there are some predictions telling that along with an increase in temperature, we're going to have variability in the capacity of the ocean to pump CO2 from the atmosphere, translated into biomass, and later sequester that carbon. So based on the characteristic of the Humboldt current system and the Chilean coast, which are a huge capacity for exchanging heat energy with the atmosphere, but also greenhouse gases, there are hotspots for sinks and sources of different types of greenhouse gases, such as CO2, but also nitrous oxide and methane along the Chilean coast, which is characterized by some of the strongest upwelling centers in the world. Along with the Peruvian coast, we can sustain almost 10% of the global fisheries, and that requires a lot of biomass and a lot of photosynthesis. But it also produces a lot of greenhouse gases. So we needed to map these hotspots, learn where they are, and also map the regions of the Chilean coast that were yet heavily undersampled. We needed to increase the information the biological information, but also the biogeochemical one and the physical one along the entire coast, if we are to characterize Chile as a globally and climatically important region for the world, as it is. It has also one of the most conspicuous and intense oxygen minimum zones, which means that below the surface, in certain areas of the Chilean coast, there are water masses with virtually no oxygen. So anoxic water masses that can last for 300 or 400 meters depth and create a particular kind of ecosystem that only exists in certain parts of the world, one of which is in the African coast, with whom we are expecting to collaborate in the future to compare the results that we are obtaining here. So we have a great climatic role because of the characteristics of our coast. We have upwelling centers that sustain marine productivity, but also create carbon footprints that need to be mapped. We can sustain a lot of global production, along with one of the most intense oxygen minimum zones in the world. We have one of the most conspicuous also freshwater reserves for the world in terms of glacier cryosphere and fjord dynamics in the south of the country. So based on this is the track of our expedition, the track of Tara along Chile. So based on this characteristic, we planned the expedition to answer the following question. Can we describe areas with climatic roles along the Chilean coast and verify if the ones that are already known are staying the way they were, or are they evolving in time? Can we detect changes in past characteristics of distinct features? Can we obtain data that, when integrated with other data, depicts the status of the biological pump? In other words, can we map the health of the Chilean Ocean? And if so, can we do it repeatedly? We based our work also in tracking the biological pump along the Chilean coast, which is also a priority for international programs such as COPAS with a K. That is a project that I think Andrea will talk about later on and in the days to come. The variability and the evolution of primary productivity, of the capacity of the ocean to pump CO2 from the atmosphere, and the features that are typical from our coast and their evolution and dynamics. So based on this, we had a group of 34 stations that were done, along with ongoing continuous tracking of physical and biogeochemical variables. In the north, we concentrated on the dynamics of the oxygen minimum zone. In the central part of Chile, of course, oxygen minimum zones, the expansion of the areas of hypoxic waters, the presence of submarine canyons that are conspicuous and that connect the rivers with the coastal ocean and can be a hotspot not only for diversity, but for biogeochemical processes. The upwelling centers along the central coast of Chile, and in the southern part, of course, the fjord dynamic and the coupling between marine and freshwater conditions. How are they behaving, and how can we predict the future of this freshwater reserve? So in all this, we spent, as I said, 4 months. We created this consortium of 9 centers of excellence that put together resources out of nowhere, because there was no, and there is still not, one single project that englobes the consortium. This is individual efforts and financial efforts from each of the centers in order to take the samples and ensure the analysis afterwards. And so far, we have been able to analyze some of the samples. You mentioned the challenge of shipping the samples abroad. Eric was in Valparaiso and also in Iquique when we did it also in Chile, and it was quite a challenge, but we managed to do so as well. And so these are the first raw results, mostly done by our PhD student here, Edson Gomez, that will be working on some of the in situ and ongoing data. So this is mostly the entire coast of Chile, sampled during the Tara CEODOS expedition. We see clearly that we are going from south to north to more warmer conditions, and we divided, our legs according to the features that I already described. The preliminary data of the stations that we sample in nutrients talk about, what we already know: very rich waters in the upwelling centers, but some interesting features are coming along as well. For instance, our data from methane and nitrous oxide on the surface of the ocean already show some interesting hotspots that we need to look further on. So in the central part of Chile, we are, outgassing probably more than we already knew some of these gases that are channeled by marine productivity and by of course, the activity of the microbiome. Nitrous oxide is a greenhouse gas that is more powerful. This is data from Laura Farias, one of our partners that just put it together last week. We know that the ocean is one of the main sources of nitrous oxide for the ocean. It is heavily has a heavy role in global warming because of its calorific effect. And the oxygen minimum zones in Chile are one of the main producers of this gas. So it's almost entirely biologically mediated. So if there isn't any high marine productivity, you won't produce this gas. But you also need low levels of oxygen, which are also present in this water. The idea here is to continue to monitor these particular points where we know that we are outgassing this gas, and also combine it with existing data from time series in key points of the coast of Chile and increasing our array of observation. We want to enhance our sampling capability and automatic going towards automatic monitoring systems that can tell us how these gases are behaving in the water. Same with methane. Some processes, biological processes in the ocean, are able to produce methane. And in some areas of the Chilean coast, we see that we are producing more than absorbing of this gas. The nature of these processes, the protagonists of it, are yet to be determined by the genomic data and the other experiments that we will do in the future. But this is a first step that we are very interested in following up. If we go north to south, we see that, of course, there is a progression, a normal progression in the temperature and salinity, but also we are tackling some proxies that we want to develop as indicators of biological activity. And this is the case of the fluorescent colored dissolved organic matter, which is one of the themes that our PhD student will tackle in the coming years. We want to create new indicators of hotspot of diversity and productivity for the Chilean coast. That might be one that can relate to changes in temperature, changes in oceanographic conditions, changes in the way of the ocean cycling this dissolved organic matter, and the optical properties that it has. So because we have the whole ongoing system for this data set, which is more than we ever were able to sample for this particular kind of proxy. We will be able, if possible, to develop some indicators of biological changes in the carbon pump along the Chilean coast. And hopefully, with the rest of the partners, to increase the comparison capability that we have. We want to work with the Atlantic lakes, if possible, with all of them, in order to compare what we obtain here and get better conclusions and results. In central Chile, this is the coast of Valparaíso and later in Valdivia, just to show you that we were interested in working submarine canyons. So you can see that in this very poor picture that I put, but this is one of the main canyons in the region, and you can see also that along with a very particular dynamic in greenhouse gases, we have the expected tendency in nutrients. There is nothing really wow, about this, except for the fact that we have the data, yeah, and we are starting to work on it. So the other interesting thing is that we see features, thanks to the resolution that we have, that we haven't been able to see before. So accumulations of greenhouse gases, the extension of the signal is something that is unprecedented for the Chilean coast. This is very raw preliminary data. We still have work to do to get the conclusions right, but it's a very promising first start. So not only in the surface, continuous sampling, we were able to have the best resolution we ever had on these kind of parameters, but the work of the TARA protocols along every single station means that we have more data to couple, along the entire territory than ever before. So the way that we want to approach this, the analysis of it is feature-based, so working on the fjords for the continuum between freshwater and marine waters, working on the upwelling centers to see the expansion or contraction of the oxygen minimum zone if we can, and in the northern zone to contrast with our partners in the Atlantic. But we see things here in the Biobío region where there's the main upwelling and hypoxic condition that we have, that we, as I said, are able to already see things that are kind of funny, and that we have never seen with this resolution before. We have time series stations here very close to the coast, with which we can contrast our results, but we can also build future cruises and future sampling strategies based on what we observe here today. So subsurface accumulation of methane, the dynamic of nitrous oxide, both powerful greenhouse gases, and the rest of the trophic chain and the carbon cycling system that will come along in a few months, I hope. So this is another example. And then when we go to the Patagonian fjords, then the work begins to look a little more complicated because of the initial resolution of the expedition. But nonetheless, there is some interesting information on dissolved organic matter that can tell us things about the coupling between the channels, the glaciers that are inside, and the marine conditions that are outside, and how they will behave in the future. So this, when we combine temperature with the distribution of colored dissolved organic matter, we see, or we start to see things that are, interesting, and where we can invest our limited financial resource in answering specific questions. So where we are now, 2 years after the end of the expeditions, there were analyses that needed to wait the end of the other legs to be done. We have these parameters that are complete or almost complete, in the laboratory analysis, and we are starting to look at the data, as I told you, because of the completion of this analysis. So we are hopeful that partial analysis that we have undergoing now are going to be completed in the following months, so we can start working on specific questions and answering the ones that we designed for this expedition. So phytoplankton and zooplankton diversity and export. This is an expedition that was designed to study the biological pump, and so on board we had sampling for zooplankton, phytoplankton, not only diversity by genomics, but also by image and export capability. So these are time-consuming samples that need to be completed. We are also studying the microplastic colonization and degradation, so the diversity of the plastisphere. There were samples all along the Chilean coast in surface waters. The samples are being analyzed in France, in Banyuls, right now, as did the nutrients that were completed last week. So also, we are heavily invested in studying, analyzing the continuous or the ongoing, the pumping surface parameters that were worked on during the expedition, environmental DNA, and the genomics part that Alejandro will give you an update, I think, tomorrow. So the challenges for the future. This being a challenging program, it still is, but we are heavily committed to make it last in time. This is not for us, at least for the centers that are present here. This is not a one-time shot. This is a program that is designed to be repeated in time every 5 years. And we said 5 years because we knew that there was no way we could do that in less time than 5 years. So we are trying to prepare the next expedition, even if it means that we will have to couple cruises in time, successfully, a succession of cruises along the Chilean coast. But we are committed to repeating the effort at least every 5 years, because we believe that this is the only way of, at least for the Chilean coast, to increase our observation capability that will later translate into predictive models. So we need to provide the data that models need to make better predictions for us. So the analysis of the extra or the other CEODOS variables need to be completed. The integration of the data, aerosol, trace metals, imaging, all of the things that we collected on board and that we need to start looking upon. That's probably the next workshop that need to be done, and start talking between groups. Daniela said it very clearly. There is this new way of cooperation in which we need to answer specific and common questions, and I think now we are finally getting underway with our part of the job for providing the data to do such an exercise. So CEODOS is, as I said, a consortium of all these people that are behind the effort. This is an effort that is, as you can see, of national implications. Almost every excellence center in the country was involved and is involved, and we expect it to be this way in the future because, as I said, this is an effort that needs to be repeated along the Chilean coast at least every 5 years for us. So funding ideas for CEODOS, DOS 2, 3, and so on are more than welcome. Thank you.
Okay, let's say I am this emotion to see data.
Don't cry, don't cry. Yeah.
So please.
So the data that you gather here in CEODOS, they depend on the seasonality of the measurements, I guess, the samples. Is the plan in the future to do samples at other times, other seasons, so that you complete the data? Or is it to repeat the same so that you have more robust data for the seasons that you already observed?
Well, yeah, that's a compromise because one of the exercises that we need to do is to couple this data with all the other time series efforts that are ongoing in the country. So there is time series ongoing in the north of Chile, in the center, in the south, that we need to inject energy and money to so they can survive, and so filling the gaps on the temporal scale. Of course, the best season for repeating this exercise is the same, spring, summer, and so on. But we are working also on opportunity cruises that come along the way using the same protocols. So the key idea is to repeat the same things that we did, in every single opportunity that we have. And this year already we will have 2 winter cruises and 1 spring cruise bi-coastal, in some of the areas. One of which is the transition between the center of the country and the southern part, which is usually an area that you just pass it because you're going somewhere else. And is one of the least studied ones. So we are trying to fill the gaps, as we go.
Yes, Daniel and Damien. Damien first.
Just to is there any other omics data from time series? You were mentioning time series that were available.
Yeah.
So are these omics data freely available for other scientists? Or, I understand that within the frame of CEODOS there will be available data. So how do you think that we could connect previous data sets?
Well, there are some previous data sets from station 18, for instance, in the center that is already available. And also we are trying to get the finance, for sequencing, ready. And one of the things that we would like to do is to enhance our capability to sequence, more quickly on site. All the centers that are involved in sequencing are committed to make it public. So the key thing is the output rate of the data. Daniele?
I have actually seen a question. So are you going to set up a regular sampling in these tools in one place or tools on observatory? Do you have a plan for that?
Well, our time series programs are running, with not all the parameters that we would like to. But one of the idea, if we get funding, is to increase the observation capability, and also to make it as automatic as possible.
The question is that if you want to empower, because the question comes from the fact that in Naples, in our institute, we decided to set up an observatory of the Gulf of Naples, which is based on this protocol. So we go out and see every month, and we go through some variants. So now we have 3 years long time series, so we can getting back to the seasonality issue. It could be useful, I think, to go deeper in one place, at least, to try to solve really the seasonal changes and all the processes with the this one. It's just to know, because
Yeah.
it's interesting to exchange.
Absolutely. I mean, there is one. I think one of the most robust time series is of Concepcion. It has nearly 20 years. It has seen a lot of changes in the amount of protocols going. because of funding, of course. But the idea is to make it more robust there with boys and also going, every month. They are still going every month, but we need to get back the genomics and all the other. And also fortify our observation capabilities in Patagonia. We have a time series in Tortel, based on physical oceanography for the moment, and a little bit of biogeochemistry. We want to make it more robust, and also doing the same in one point. It requires money, and it's a very good thing that we have centers, that are working for this with a budget that is consequent. But the more, the better. So we are always looking for funding for these kind of things.
Okay, so since we have, so Andre told me, we have after a streaming talk, so a Zoom talk at 11:45 exactly, so we will maybe leave the questions for the week and the coffee break. So we have a coffee break now of, let's say, 20, 25 minutes. Yes. In any case, just some news. We have, in fact, Iñaki sent me yesterday, almost at 3 a.m., some slides about the taxonomy, or bacterial taxonomy of Chilean code. Is the first description. And also we have a sequence and assembly, thanks to Damian and Dante. We are the founders of this part. The prokaryote part, of not all the prokaryote, the bacterial part along Chile. So I think that we will be able to, at least to discuss with this preliminary data, integrate with nutrients and physical data very, very soon. I think in the next 15, 1 month, I think the data will be released to the consortium.
And also with other databases that have been collected for the last 20, 30 years, in key areas. So the thing is to pass from key areas along the Chilean coast to a full array of data integration and observation. Let's have a good coffee.
Yes, this is coffee time, so thank you very much.
Yes, do you hear me? Okay, perfect. Gracias.
and Romy for being here at this workshop. So essentially today is focused on science to policy issues and the way that some French agencies in particular are thinking the ocean and the challenge of the ocean. So first will be with us Cyril Barnerillas from the OFB, and the title of his discussion is EBAs, EBSAs, and existing monitoring and policy tools. So welcome, Cyril. Thank you for being here.
Thank you very much. Thank you, André. Hello to everyone. Sorry to be with you, of course, today. I would have loved to be here instead of being another rain in Paris, but that's the way it is. So, yes, first I would like to acknowledge, of course, my colleague Fenia Mara, who helped me in preparing this presentation. So, sorry. I will first go through this question of how to consider marine ecological connectivity into the implementation of environmental international conventions, like multilateral environmental agreements. And so I took this, I think the idea and the concept of marine connectivity is quite important, as you know, when we consider marine biodiversity conservation. Indeed, it is well taken in charge by the Global Biodiversity Framework, which is the agreement that was taken under the Convention for Biological Diversity in December in Montreal. As you can see here, it is of course very important for the flow of natural process, and all the more under the current climate change crisis that we're going through, as you can see here. So when we speak about the science to policy interface, we can. So I think it's quite important to make to have a clear distinction between what is science, and it's quite important to let the scientists do the job to be the most neutral possible, and the policy work that can feed on this and that should feed on science data and science work. So here on the left, you can see some examples of, and I will go deeper into some of them. Some examples of scientific activities that have been done on the marine environment,
like IBAs, important birds areas, important marine mammals areas, EIMMAs. And some coming new ones that are IMTAs for marine turtles and ISRAs for sharks and rays. So that altogether can help building KBAs, so key biodiversity areas. And in the marine environment, you may have heard about EBSAs, so ecologically and biologically areas that are designed under the Convention for Biological Diversity. And here, that's where we see that when you are not in a totally scientific process, it can be very difficult, because under the CBD, the discussion for around EBSAs have been quite difficult, because some countries have decided that EBSAs could represent some political lines or borders. So the discussions around EBSAs are always very difficult. And on the policy side, you have the marine protected areas, of course, and the other effective conservation measures, OECMs, that are ways to protect conserve biodiversity. That should be part of MSP, Marine Spatial Planning. that should be done in a regional governance framework, of course, and that can be linked to the global biodiversity framework that I talked about before, and also to the BBNJ negotiations. BBNJ standing for biodiversity beyond national jurisdictions, which are the high seas, what we call the high seas, so beyond EEZ, beyond the economic exclusive zones of countries. And this BBNJ treaty has been dealt finally after 15 years of negotiations in New York last March, and now it has to be ratified by countries before entering into force. So that's the EPSA process that is presented here. So normally, I mean, it's based on regional workshops, research, and allowed. I mean, made possible also by capacity development for all countries. Then the CBD Secretariat, so the Convention for Biological Diversity Secretariat take care of the EPSA data repository and present to the CEPSA the data that is then submitted to the CBD COP, where you have the political discussions coming in. When EPSAs are agreed, they then get officially recognized, and you can see here a map of EPSAs that is also available online. Here you can see you can see a zoom of EPSAs. So that it is a fruit of 15 regional workshops, and candidate EPSAs have to be described by scientists of relevant countries or more countries than one if needed, based on 7 criteria. So nowadays we have 321 EPSAs that are approved, and 17 coming in, especially in the Northeast Atlantic, that are awaiting approval by the Conference of Parties of the CBD. So here you have a small snapshot of what EPSAs represent. You can see that almost half of the EPSAs are in shallow water. I mean, shallow, less than 200 meters deep. Some are very big, like the North Pacific Transition Zone. And so you can see that, and you can also see in the Northeast Atlantic the proposed EPSAs that have to be agreed, considered, and agreed by the COP. I will present other, examples of scientific processes that can support decision making, like IBAs for birds, IMMS for marine mammals, and ISRS for sharks and rays. So the data concerning birds is quite important, as you can see some snapshots here. The process of delimiting IBAs has been supported especially by BirdLife worldwide. So you can see here that we have, quite a lot of data. Of course, you still have some zones that are less covered, like on some oceanic without islands zones. That represent 25 million points, quite a lot, 150 species, and a lot of data contributors. So that is also one key point. So you can either have data coming from from birds that are observed directly on the ground, or also by birds that are tracked through GPS data positioners, for example. For you can also I mean I'm happy also to present a similar work that has been done for marine mammals that allow to produce important marine mammals areas, IMMAs, that you can see in the bottom map in orange. So it has been especially supported by Gobi, by the Federal Ministry of Germany, and so implemented by Gobi and Thetis. So they are very, I mean, some of the criteria that are used are the vulnerability, as you can see here, distribution and abundance, key life cycle areas, and distinctiveness and diversity as special attributes. So the idea is to compile data of of many sources, and very often when you build an IMMA or an IBA workshop, you bring together scientists that have worked on the same group of species and in the same area, which can be like the subantarctic zone, for example. And they share the data, they share their way of working, their hypotheses, and time after time they can draw some areas of interest, as you can see here on that is the first stage, and then candidate IMMAs, and the final stage is confirming the IMMA that should be kept, and that can serve afterwards for advocacy and for preparing for policy decisions. So here you can see different type of zones that can be used. So we mentioned EPSAs, for example, and KBAs. I will continue now with important sharks and rays areas. So the idea is to base also conservation on this group of species that are quite fished and also not very well known in terms of conservation status. So the first expert-driven and classification of habitats has been organized recently, and the idea was to ensure that sharks could be incorporated in ongoing conservation planning processes. The idea, of course, from the policy implementing agency point of view that I represent, is to reduce the mortality of sharks and rays that are very important, as you know, of course, for the conservation of biodiversity. So here is the guidelines that are being produced by the shark specialist group of IUCN Commission in August 22. We can share the reference afterwards, of course, for those who are interested. So here is an example of the work that has been done in the zone of that is, I mean, closest to you, in the southern Eastern Pacific. So in this workshop, 74 candidates ISRAs have been proposed, and 5 areas of interest. So you can see here the process of creation. You often start with closer zones that people know better and agree on easily, and then you can expand on other zones when you group data, as it is on day 5. And then you can cross the candidate ISRAs with existing marine protected areas, and you can see the gaps that exist, or sometimes the good coverage. For example, in the Revilla CEODOS Islands. But you can see, of course, many gaps that could be covered in terms of policy implementation. Here is, I mean, some references of also of the use that can be done of key biodiversity areas, so of scientific data for policies. In the link, you can see quite more of them, but I just wanted to pinpoint some of them, like the work that has been done on the potential harm of World Bank project that can be done to area reach of biodiversity, the use of IBS for their impact on biodiversity, on conservation policy, advocacy, and action, and also the cost of meeting the global biodiversity targets that has been done using the KBA's data. And I will conclude by focusing on the work on important marine mammals areas in the Southern Ocean and highlight some of the work that has been done to bring the data to the CAMLAR committee. So here is a focus on the Southern Atlantic Ocean and the important marine mammals areas that have been designated by scientific data and scientific work. And you can see also the candidate IMMAs in red, that have also been kept in case. The report is also available. And I will go directly to the work that has been done using this scientific data to bring it to the scientific committee of the CAMLAR, of the so the conventions that regulate and protect living resources around the Southern Atlantic. And it was very important to, in this quite difficult politically speaking context, to have very robust and neutral scientific data to propose them to the scientific committee. And I will stop here. Thank you very much. Romy, I think the floor is yours.
You can speak loud.
Yeah. Salut Cyril, merci pour cette super présentation, très utile. We were planning my intervention that you hear from me in the afternoon, and after this focus. But as in French, it is already late. It is 6:30, so we spared our friends some hours waiting. So this presentation, but we will develop this policy part in the afternoon. I will do some kind of state-of-art presentation in the beginning, and then we will hear from the ministries in Chile also. And just want to say that this hill is a kind of also one of the inspirations for the COPAS projects, the key ocean planktonic areas. When we started to think about how we could design a plankton hotspot MPA, and Cyril was just arriving from Washington when he worked in the US also, and he said there's a nice way. There's a KBA, which is an existing frame that exists for biology, and I don't hear about an ocean KBA. So we started to explore this opportunity to have an ocean KBA, and thanks to Cyril, just to remind our conversation in the Tara boat, which gives birth to the idea to go to an ocean KBA. Thank you, Cyril.
Thank you, Andres, for the context. So the next talk, thank you very much, Cyril, is by Romy Ettinger.
Yeah.
And So Romy is maybe you're better to. She's the coordinator of the AtlantEco. Yes.
Yes. Yeah. So Romy will explain the project. Hello, Romy. Bonjour, Romy.
Bonjour.
From this Mission Microbiome and the CEODOS designing and this idea to have a KBA for the ocean, we wrote a project for the French Facility of Global Environment, and the project was approved. So now we have some nice resources to go through it. We have the time and resources to work on this ocean KBA. And also another chapter of the AtlantEco will be improving fish prediction tools, also with some African partners. So Romy will explain all of this.
Thank you, Andre. Thank you, Romy. The floor is yours.
Thank you very much. Hello, everyone, and thank you for the invitation. It's a pleasure to make this presentation. As Cyril, I wish I would be here with you, but you have Andre, and I think it's great to be there. As Andre said, the project is starting this year, and we've been working on it for 2 years, working on its building for 2 years with different partners from France, Chile, and Senegal. I want today to thank the CMM and especially Alejandro Mas for his support and commitment to our side to make this project possible. So it's always a pleasure to work with you, Alejandro. So thank you very much for that. So I'm going to start with the context and the objectives of this project. So it will be conducted from 2023 to 2026, and it will be officially launched in June. So it's a lot of work and challenges ahead with this project, but we are very proud to conduct it, and it's a very innovative and ambitious project. But first of all, I want to say that it's not only about science, but it's a science to policy project. This is really important. It's also a scientific cooperation project, and it's about making science on plankton and microorganisms operational to guide decisions, political decisions. So the global objective of the project is to contribute to an integrated management of marine ecosystem by increasing the knowledge, but sharing it with decision makers and also general public. I will explain later how the general public and even kids are integrated in the project. So, and this project arrives at the right moment because the science will have a fundamental place in the future international and regional ocean governance processes. We need you scientists for different processes. We talk about BBNG, but also the CDB. There will be impact in 2027 in Senegal. So a lot of issues we have to address during these governance processes, and we need the science, we need the data, and we need also science to policy tools. So we're going to talk about this during this presentation. That's it. Yeah, I just want to add, yeah, when I'm talking about we need scientific tools, it's, for example, working with the scientists on new criteria, on new methodologies, also on science-based management tools to identify, to predict, to better gestion and monitoring of the ocean. So it's a lot of work to do with you. So we do this job with different partners at the international level, but also regional level and local level. So as I said, Chile, Senegal, but also Mauritania. So I'm going to talk about these great partners. First of all, the co-founders. So we have the financial support for the French Facility for Global Environment, the FFEM, who is not here today. we have the sorry, the French Biodiversity Agency, the OFB. So thank you very much, Cyril, for the support of your institution, and also the French Environment Ministry. Then we have the scientific partners. So we have the French National Research Institute for Sustainable Development, the ERD, which is very well implemented in Africa. We have the Laboratory of Villefranche. We have the University of Nantes. We have, I'm going to try to say it in Spanish, the Centro de Modelamiento Matemático. Sorry for my Spanish. And also we have other partners, Plankton Planet, which is an institution working on participative science on plankton. We have, of course, also the AtlantEco project working with us. And also we work with partners at the regional level, like the PRCN, which is the Regional Partnership for the Conservation of the Coastal Marine Zone, an NGO, maybe they, doing a great work on the field with kids and also education partners, and the Bacomab Trust Fund. So during this project, we also work with other indirect partners and actors of interest. So you can see Ideal, CEODOS, of course, the Genoscope for the scientific part. We have the EUCN, but also we want to work with regional fisheries institution and the local Senegalese institutions and networks of MPA in the Western African region. So a lot of people to work with. Then I'm gonna do a short presentation of the 3 components and activities of the project. So the first one is the science. So the project will have 2 axes of research. The first one is the development of a configuration plankton to fish of the APECOS model, which is a fishing model which already exists, and to work on it at the Senegalese Mauritanian operating scale. So the objective of this work is to better understand the fishing efforts in the region, but also to have a better prediction of the evolution of the stocks, taking account all the ecosystem. So this work will be conducted by the IRD. And then the second axe of research is the development of a new methodology to identify key ocean planktonic areas. André was talking about it just before. The methodology is to make the match between satellite data and genomic data, data from previous Tara expedition, the Tara Ocean expedition, but also new data from the program CEODOS. And to the objective of this work is to develop a map with the identification of zones with climatic interest based on the study of plankton and microorganism data. So the objective is to make this tool as a reference to evaluate the health of the ocean and to have to be able to have new data and new criteria to explain the ecosystem services of the plankton and microorganisms and where these ecosystem services are the most important in the ocean. So both of these research will work on upwellings on the Chilean part and the West African part. And the idea of this project is that these 2 axes of research will interact together, because this research can interest both each other. So try to create this cooperation between the different teams in this project. The second component is the capacity building activities. So it's to set up a training courses and imaging platform in Dakar in a Senegalese institution. So with different 3 types of equipment, the ZooScan, the UVP, and the plantoscope. This activity will be led by Villefranche, the laboratory of Villefranche, and the RRV. And the objective is to develop the different research projects at the level of the West African region on planktonic ecosystem, because the research on this ecosystem the projects are lacking, and the institutions would like to work more on this part of the ecosystem. So, and this working on that kind of this platform will help to have different kind of project that would be very important to address some local issues, coastal issues, especially, for example, for fishery predictions. And the objective of this also is to enlarge the community of scientists working on plankton to study this ecosystem, that try to have a multiplication of the projects all over the world, and especially in Western Africa for this project, to be sure that the scientific communities of the states are sharing the same knowledge on this ecosystem. And then the third component is the outreach and advocacy activities. First, outreach is we're going to work with Nebedai, an NGO, and Plankton Planets. So in September 2022, we were in Dakar with the boat Taha, and during the stopover, we share great moments around special observation and discovery equipment that Plankton Planets created. And we had animations with local NGOs, and also other general public. So it was a proof that you have a great local interest in Senegal for learning and to see and to have the possibility to watch the incredible world of plankton through these different equipments. So the idea of the project is to go further with this NGO and have participatory science, citizen science with NGO with this different equipment. Then the advocacy activities. So it's André and I part. So as I said at the beginning of the presentation, is our objective is to share your work, the work of scientists, to share the knowledge. And during this project, we can almost talk about not only advocacy, but advocacies with S, because we as I say, we will have different scientific tools, and we have to work on different scale and to talk about the different tools in different processes. So we were going to work at the regional level, for example, with the work on plankton to fish configuration of the APECOS model. We're going to work at the international level as for the COPAS methodology during BBNG, CDB. We're going to have a great moment for the ocean in 2024 in Costa Rica, but also in France, in Nice in 2025. So it will be great occasions to share the work of our scientific partners. And there will be a lot of subjects to address during these processes, but the Taha Ocean Foundation will keep on working and staying and also defending the place of the science, the importance to have an open science, also an inclusive science, to foster cooperation with Latin American countries, African countries, We will also be mobilized with the scientists, the definition of the levels of protection for the high seas. Once the treaty will be ratified, be sure that the scientists have the place in this treaty, and also working on gestion tools with all the work which has been done of the area-based management tools. So for this part, the objective is to have scientific inputs during the processes, and also to structure the scientific information. Be sure that plankton is going to be taken into account by decision makers, which is not enough the case right now. It's pretty much it, and I'm more than happy to answer your questions if you have some. So thank you very much for your attention. I just forgot this slide is to show you that how we'll do the advocacy work is organizing side events. It's also to write with the scientists policy briefs that we will promote during the conferences, or even different committees or important ocean governance time. And also working on mapping tools. For example, just put a capture of MapMaker, which is a great tool that the USCN developed. It's an example of what we want to go to. It's science to communication tools for decision makers that make all this science understandable for decision makers. And that's it. Thank you very much. Yeah, you're very far away, but it's normal, I would say.
So have you defined already if the criteria for these zones are species identity, diversity, or ecological processes or functions, or is this something to do during the project, or is already defined?
I can't hear you very well. Sure. Yeah.
So the criteria to create these zones, is it?
You're talking about the key ocean planktonic areas?
Yes, yes.
Yeah.
Economy or species identity or diversity, or is ecological processes or ecological functions? Is that already established, or are you going to work on that during the project?
We're going to work on it during this project. There are Alejandro and Damien with you, so they are the experts. So maybe I'll let them answer the questions of the different steps to develop the methodologies.
Processes using the genes. So the taxonomy will come on. So we will use the biological processes, and we want just to map the biological processes. So basically to link what we could learn from the genomics from space.
Great. And some other ideas that
but that
come
starts
during
the
sure.
I think that essentially is open because there are some.
So the start is this, and then
Papers from
and then
this is a starting point, but now we have other methods and other ideas. So I think
that's cool.
This is an important part.
Very interesting. But it's basically functions and processes that we want to measure rather than species, richness, or classical indicators.
Notice the dynamics. In fact, the idea is to see if can we put some dynamics. This is a question from people in SFM. It's a complicated question, by the way. Can you put some dynamics in the key ocean protected areas?
or not?
If the areas move?
Yes.
Oh.
This is an important point, but in fact it's a complicated point.
Yes.
Or seasonality.
Yes, because sometimes the law, the law will say this, but maybe you can see that it is moving. Camila will go and take some measurements, and we'll see that this is here now. But this is an issue, that's why it's called a methodology. The idea is to think in a methodology to, I don't know, how to propose something that is dynamics, but it's not too basic. Because FFM asked at some point, if I remember right, that maybe you will finish with all the planet.
Yes.
Completely protected, and nobody will want that. Maybe us, maybe Antoine, I don't know. We. No more questions. But if I see that, yes, please.
Just a brief one. So, who is funding this? I mean, I got lost with
this. This is a French. It's Fonds Français pour l'Environnement Mondial.
Okay.
It's a French agency for, let's say, for policies. It's not for. It's not for science. It's, the science will be here just to support the group of people working in policies. In fact, this afternoon, Damian will be here. Maybe he can tell us some ideas that he's developing with a lawyer. Yes. Just trying to see how you can mix science with the. This is novel ideas, in fact, crazy ideas. How to make a law from.
It's important to say that it's an innovation project.
Yes.
We are here to make a proof of concept. So this will be building the methodology, and we hope to, in 3 years, to prove the feasibility and the relation with data in this project, to start to propose, as Cyril showed, to propose to the UN system. There's a whole stuff.
Connect the job with smartphone connected.
To propose an indicator, a new tool in a global basis, or even in a regional basis, we have a long way to go. You need to lobby, you need to work with the ministers, you need to propose a first experience applied, and then maybe in 5 years and in 7 years, the CBD will be including the COPAs in these KBAs that are indicating to the governments where to protect. The idea is to do this way with the policy team of the process.
By the way, what I think is it could be completely dynamic also in the sense of markers. Maybe the first version will be this kind of markers, but if the science goes beyond, maybe other markers can go inside other measurements. The point is how to make the link. Propose something that is reasonable with good indicators. It's more econometric thing. Yes.
We'll speak about that in the afternoon.
Yes. Okay, thank you very much, Romy, for this.
Thank you very much.
I see that Andres is hidden behind, but not hidden, but I don't know. Since Andres is here and he helps a lot to this expedition, maybe. And he was in policy, in fact, for many years, some years. But now, Andres Cubic, was former Ministry of Science. Andres was also now he's working, trying to make links between policies and science and in different issues. You can correct me, Andres. Yes. Maybe you can give us some view, maybe of what are you thinking about policies and science. Maybe we have time, maybe 10 minutes if you are not prepared, maybe, but you're always prepared. Huh? But since you're here, I think we can use the experience of Andres maybe to discuss, because Andres needs to move this afternoon.
In the behalf of the Tara Ocean Foundation, I'd like to thank Andres officially in the name of the foundation for all the efforts you did during the expedition. That was crucial. Thank you.
First time you can do that.
So thank you very much. I'm not prepared at all for the challenge, yes. which is a mighty challenge, right? And maybe two things which I think are worth taking into account. The first one is that public policy and politics moves
in a very non-linear way. That's the first thing. And so you have to tackle it from many different angles at the same time. This is something that I've learned, and it's something that I think Alejandro was talking about a methodology. Well, we need to develop a methodology in which you tackle this issue by multiple fronts at the same time. Having said that, we really would like to have all these linear processes to be non-linear processes, at least very objective, and they are not. So they depend a lot on the people. And so I think I'm going to put you one example, which I think is a great example of how science is moving into policy in Chile and abroad. There's something, I don't know if you've heard about it, it's called neurorights, neuroderechos, which is trying to protect humans from the information that devices can obtain from our brain activity. This was something that came out from a lab at Columbia University by Rafael Yuste. He came to Chile, he's a neuroscientist, and he proposed that it was time, even though we didn't need it right now, to protect the information that's coming out of our brains and that are being registered in screens and medical tests and by devices, etc. He became very close to a Chilean politician. They arranged a series of meetings in the country where the idea was promoted with scientists and with the general public. This went on for about 3 years. After 3 years, Parliament started to develop a policy about protecting neural rights. In 3 years' time, it went into our constitution. It's now in our constitution, the current constitution, the protection of neural rights. It was used as a pilot study, as a proof of concept, and now it's moving internationally, and it's reaching the UN. And it's reaching the UN, and they are trying to establish this as a fundamental human right. So it went from some ideas in the US to a proof of concept in Chile by a Spanish neuroscientist, and now it's going to the UN. I think it's a very successful way of how policy at national and international level is being made on a topic which is state of the art, which is cutting edge knowledge. We may agree or not whether we think this is required today, but I think this is something that it shows us how this thing moves. I think you need champions, both in the scientific fields and in the politics fields. These are people, they're not institutions, they're not, they will change, but you need champions in both fields. You need a proof of concept in a place where this thing is accepted as something viable. And then you need the international connections to move it to the next level. This is a case which we've been sort of revising, which I think it shows and it tells us and it teaches us how these things move from science to policy, national and internationally. Again, you may agree or not that this is something worth doing, but this is what's happening. That's one thing. The second thing is that in my experience, any organization, whether it's a company or a university or a non-profit organization or an exploration like yours, if you're doing cutting-edge research, then you're going to need a group of people fully dedicated to policy, because the regulations will need to be changed. And this is something that even for a startup that's doing work on some novel aspect of biology, the topics that we'll address will need new regulations. And so I think it's super important that that a group like this really links to the policy aspect of it. because you're going to need to change the regulations. For example, to do things in Chile, we need to change the governance of ocean research. It's currently under the Navy. It's not currently under researchers. And that is because the decree is from 1971 and it hasn't changed in whatever, 50 years. And so it needs to be changed. And that requires a very close relationship to public policy. And it's something that we need to develop how to do it. The third thing is that I heard about ways of getting into decision making. There's several projects in Chile which are trying to do this. And one of them is a project which is going to be launched next week from Universidad Católica, linking the research fields, different research fields, to the needs of policymaking. It's going to be the first time that's going to be done systematically, so that policymakers, when they need information, they will know who to access. This is something interesting. I think it's something worth keeping sort of under our attention. Yeah.
I
can ask a question.
Yes.
Now, you were part when the laws of the ocean start to be discussed, and also the laws for the organization for the ocean.
Yeah.
So maybe you can tell us, well, you know that because you were
Yeah.
there.
So
the new roles of the institution.
Yeah. So this decree, which is 1971, we thought it was important to change it, but this requires, again, non-linear, long-term efforts. Currently, the Ministry of Science is involved in the ministerial committee. So we included the Ministry of Science in the ministerial committee for ocean research. And in the program of ocean research and ocean development, now there is one chapter, which is chapter 5, in which section 5.2 says that the governance needs to be changed. So we advanced one step. This is the ocean program of the Chilean government, which already includes that we need to change the governance of ocean research. It hasn't been done, but it has been stated that it needs to be changed. So the next move is now to include the change in some form of decree. But this is what I mentioned about nonlinear, people change, and these efforts need to be sustained. They need to be sort of carried on in different governments where turnover is very high. And so we need to focus on what needs to be done in the long term. We did what we could, our part, and now it's time to do the next thing. And it's ready, but now it needs to be done. But now the Minister of Science is part of the decision makers, which wasn't before. And one more thing that I'm going to mention, relative to the previous talk and our conversation on the coffee break. I'm sure that you're all aware with the concept of natural capital, of ecosystem services. We've been discussing this recently. The Chilean government is very strongly involved in developing this concept today. If you're interested in ecosystem services, natural capital, and how it could integrate new regulations, I think key people are today in the Ministry of Finance and in the Central Bank. Both organizations have teams which are fully dedicated to develop the concept of natural capital in the Chilean economy. So again, if you're planning to incorporate a new model into assessing the value of biodiversity, in this case, the ocean biodiversity or plankton produced biodiversity, then it's good timing in Chile. There's people dedicated to that. I think they might be more focused on land economy than on ocean-based economy. but I think it would be interesting to talk to them and see what they're up to. I'm not aware of what they're doing right now. We have a natural capital committee, which is formed by the central bank, the Ministry of Finance, and the Consejo de Ciencia, Tecnología e Innovación. And so I think there's ground there for very productive conversations. If you can establish yourselves as experts in providing the evidence, not just the biodiversity evidence, but how you value that biodiversity, then you'll become very key players in the future here in Chile, I think. And you might be working together with people that are doing the same on land and forests, which are already making some progress. So I think that could be an avenue of interesting conversations. Maybe we can try to have them before you go back to France. I think it would be really helpful.
Thank you very much.
You're very
welcome.
welcome.
A lot of advice.
And if you want to talk about these things, I'm not very happy.
Thank you, Andres. So now there is a break for kind of lunch. Time for lunch.
Have a great lunch.
Thank you, Omi. I cannot see you now. And okay.
Cyril left. Cyril left. He had to leave. So
Okay.
thank you very much. Enjoy your lunch. Bye.
Ahí está.